Hello and welcome to today's webinar. My name is Jennifer Caveney and I will be moderating today's presentation. All right, so let's go ahead and get started. Our speaker today is Dan Berg, who is an Analytical Services Manager for Eurofins Food Chemistry Testing in Madison, Wisconsin. Dan, we are looking forward to your presentation, so please begin whenever you're ready.
All right, well, thank you, Jennifer. Welcome everyone to this webinar, Understanding Protein Analysis for Label Claims and Characterization. Sure, we're advancing. There we go. Let me just provide a little bit of an introduction of myself. I'm Daniel Berg, Analytical Services Manager for Eurofins Food Chemistry Testing in Madison, Wisconsin. My background, I have a master's in food chemistry from the University of Wisconsin-Madison. I've been with this particular lab for the last 14 years. And previously to that, I worked in about 15 years in the food industry in a variety of roles in quality assurance, R&D, and technical. I specialize in food, beverage, and ingredient nutrition and contaminants. In my role, I really enjoy helping our customers find the best practical testing services for regulatory and development needs. So what are we going to learn today? We're going to start with a primer on proteins and amino acids. We're going to cover crude protein methods, as well as non-protein nitrogens that may be measured within those crude protein methods. We're going to discuss protein claims, especially from PDCAAS, and we're going to finish with some targeted protein analysis and other protein characterizations. So why test proteins? There are many reasons to test proteins, and I hope that my talk today will provide some context to how the methods differ and how to address the complexity of protein analysis. The market for protein products is very strong, ranging from traditional foods, keto diets, dietary supplements, and now supportive diets for GLP-1 type weight loss and diabetes management. The need for alternative protein sources is also very strong, with water, land, and CO2 sustainability concerns for meat and dairy proteins. There's lots of opportunities, and chemical analysis can be an effective part of the development and maintenance of successful protein products. So let's start with a primer on proteins. Proteins are the building blocks of the human body and all other life as well. They regulate how our bodies and our cells functions. Proteins make up muscle tissue. They provide structure in tendons, cartilage, skin, hair, hooves. They function as enzymes, as hormones, antibodies, and the function of the cellular membranes. Some proteins are really stored in seeds and milk as nutrients for the next generation. Now, proteins are chains of amino acids, and that functionality arises from the specific shapes that they form due to primary, secondary, tertiary, and quaternary structures. If you look at the proteins, those helixes and sheets and the folding and bends and the bonds within the protein and within other proteins and associated non-protein compounds such as glycans, lipids, and minerals give it its structure and its functionality. Think of the iron and hemoglobin. It's a very specific structuring and a mineral that has a functionality in holding oxygen in your blood. The shape and interactions are really dependent on the differing amino acids that come together to build the protein, or in our cases, we'll take the protein, break it apart into the amino acids to test for it. And we're going to talk about all these things. So let's start with amino acids. Amino acids generally have this structure of an amino group and a carboxylic acid group. So it's an amino acid. And they have differing side chains that provide the difference from the 20 primary amino acids for food nutrition. There are several other common amino acids, and actually there are hundreds of other related amino acids, each with differing side chains that may have electrical charge, they may have polarity, they may be nonpolar or hydrophobic, or they may be able to form a disulfide bridge like a cysteine sulfur group. These differences provide the structuring and allow the enzymes, membranes, contracting muscles, etc. to work. Now, proteins are composed of amino acids, and all amino acids contain nitrogen. The fact that proteins contain nitrogen and other macronutrients like carbs and fats generally do not allow for methods to measure for protein going back into the 1800s. The nitrogen content in amino acids varies, as in this example of 3 different amino acids. Phenylalanine has just 1 nitrogen and a relatively large mass from its phenyl group. Lysine has 2, while arginine has 1, 2, 3, 4 amino acids. Now, a crude protein measurement measures for the nitrogens in those amino acids, and it typically applies a factor of N times 6.25, so 6.25 times the percent of nitrogen to give us a protein value. This is based on an average of 16% nitrogen. And if you measure all the nitrogen, or measure all the amino acids together in a protein, it'll average somewhere around that. But that's an approximation. Each individual protein will have a different true percent nitrogen. And there are some specific factors that are established, such as N times 6.38 for dairy or 5.55 for gelatin. This means that dairy proteins have amino acids that are relatively lower in nitrogen content, while gelatin has more nitrogen per gram of protein. Let's talk about peptide bonds and the conversion of amino acids to a peptide and vice versa. When 2 amino acids come together, they're going to form a bond between the carbon of the carboxylic acid and the nitrogen of the amide group. And when they form this bond, they're going to lose a hydrogen and a hydroxyl. So they're going to actually release a water to build that bond. In reverse, if it's a dipeptide or a longer peptide chain, that peptide bond is going to require the addition of a water to hydrolyze the bond to form 2 free amino acids. So there is a difference in the mass of the amino acids as free and within a peptide. That mass is 1 water per amino acid. So if you measure the protein and you compare it to the mass of amino acids released, there is a difference. This is the dehydrated form of amino acids within the protein or peptides. Now, crude protein is typically a measurement for nitrogen. Kjeldahl technique is generally the standard for protein analysis. And briefly, within the method, the sample is digested with sulfuric acid to break everything down into ammonium, carbon dioxide, and water. We neutralize the ammonium to form ammonia. This ammonia can then be distilled and captured again and converted to ammonium, and then titrated to give us a final report of percent nitrogen. Now, the standard methods contains a lot of harsh chemicals, kind of some safety issues, makes it somewhat difficult, but a lot of this has been automated through instrumentation to allow for a simpler, more efficient digestion, distillation, and titration. Second is the Dumas combustion. Now, the Dumas method is a quicker technique that can be just as accurate. The amino acids are combusted in a pure oxygen, 900 degrees Celsius or higher, producing water, carbon dioxide, nitrogen gas, and nitrogen oxides. There's an online process to reduce the oxides to nitrogen and separate the water and carbon dioxide, and finally, with a thermal conductivity detector to measure nitrogen. Regardless of either of those 2 methods, the nitrogen is converted to protein, most typically by a 6.25 factor, and we'll describe this further in upcoming slides. Proteins can be measured by a number of other means. For example, colorimetric methods. Just a couple of common examples. The Bradford method is a complexing of a dye with the carboxyl group of proteins and allows for a calibration curve based on that absorbance. The Lowry method is a reaction of copper ions with proteins' aromatic residues. These methods are very accurate in pure solutions and have advantages for their simple use, but there are some limitations within the food world. So when dealing with varied food matrices, we're generally not using these type of methods. More typically, those type of methods are used in biological or basic research. Also worth mentioning is NIR, or near-infrared analyzers. This is based on a spectral signature of light that is reflected from an analyzed sample. Within proteins, we have those amino bonds, at nitrogen-hydrogen bonds, and those will have a certain wavelength will be absorbed. And you can establish calibrations based on the concentration of, or the extent of those wavelengths, very, very similar to a crude protein analysis. Now, within the NIR calibrations, we're not only looking at amino bonds, we're looking at oxygen to hydrogen, carbon to hydrogen, all kinds of bondings, and it allows for really a complex associations of the spectrum to be able to calibrate concentrations of fat, carbohydrates, water, proteins, and many other components. Now, our lab doesn't test by NIR as a primary method for protein, but our primary methods are regularly used to establish the calibration curves for NIR. This allows our customers to test in production in real time in a matter of seconds to get data that can be linked back to original official methods. Now, in most foods, we can test for nitrogen and directly use that value and report it as grams of protein. But often within dietary supplements and some foods, they will contain ingredients that will make this measurement inaccurate due to non-protein nitrogen, or NPN. Other nitrogen sources could be free amino acids, creatine, phospholipids, urea. Many different things contain nitrogen. And within those standard methods can be counted just as protein, just the same. Now, we can correct for non-protein nitrogen in a few different ways. First is by a direct analysis. So if we know that there is free amino acids, creatine, phospholipids, carnitine, etc., we can measure for those by a standard method for the individual component. We can then consider the percent nitrogen in that component and subtract from the total combustion or killed nitrogen. Alternatively, we can do a non-protein nitrogen analysis. Now, in the non-protein nitrogen analysis, we utilize the precipitation behavior of intact proteins. We're going to add trichloroacetic acid, which is going to cause the proteins to precipitate. We're going to filter out those proteins, and the still soluble non-protein nitrogen can then be tested by a Kaldol-like technique to give us a final amount of nitrogen as non-protein nitrogen, which can be subtracted from the total nitrogen for a corrected crude protein. Now, third, we could directly measure for some proteins. If you do a direct measurement, you can avoid the non-protein nitrogen. And I'm going to cover some specific protein measurements later, but a direct analysis of complex foods has many limitations. So this option is really only relevant to some more pure isolates. The last option is to test directly for the amino acids from the intact protein. So we can do an amino acid digestion of the protein and measure for those amino acids, just as I'm going to describe for the PDCAAS score. This will give us total amino acids, which can be corrected to the theoretical intact protein by subtracting any free amino acids and by correcting for the dehydrate mass of each amino acid in the protein. In this case, you can ignore the non-protein nitrogens as you have a more direct, specific measurement of the amino acids in the protein. Also related to non-protein nitrogen is adulteration. Remember the melamine scares? Thousands of babies sick in hospitals in China, and then a scandal spreading globally. Those events really made everyone realize how vulnerable a supply chain can be when it relies on a really simple protein quality method that doesn't have the specificity that may be needed to have a thorough method. Take a look at the melamine structure. The molecular structure has 6 nitrogens in a relatively small molecule. It's a perfect source to boost the nitrogen content in milk powder with little notice to any other effect. I'm not going to focus on adulteration today, but the melamine situation was a major catalyst to food safety initiatives related to supply chain integrity and risk from economically motivated adulteration. We have analytical methods to measure for melamine and other nitrogen-rich adulterants, but for any product that has a simple quality marker, it's worth evaluating secondary means to confirm its authenticity, especially in times of supply chain stresses. For example, tariffs or availability issues, risk for this may grow. Now, several proteins have monograph methods, which may include multiple identification methods and quality criteria to confirm its purity and identity. But if monograph methods are lacking, there may be other orthogonal approaches that are valuable to find unexpected issues. This is where we'll test for multiple parameters of a food and look for differences, which may or may not be related to the protein content, but are indicative of a difference from authentic. If I were to test an adulterated milk with melamine, I may be able to detect that difference directly from a non-protein nitrogen test, or a test for the total amino acids, or even looking for things like lactose, calcium, and phosphorus. Those may provide clues that something's different depending on how any milk solids were replaced. Orthogonal authenticity approaches include testing for organic acids, amino acids, minerals, sugars, etc. Are very common in fruit juices, honey, vegetable oils, and other commonly adulterated foods. But techniques can also be adapted to protein sources as well. Now let's touch on some other special considerations with measuring protein by a nitrogen analysis, especially with novel foods. The first example I'll talk about is chitin. Now, chitin is a polysaccharide. It's present in insects and crustaceans. There's more interest in new sources of protein, such as insects. And chitin is actually the second most abundant biopolymer on Earth after cellulose. Now, it is primarily made of N-acetylglucosamine. So what is that? All right, so N-acetylglucosamine is a glucose molecule that has an amide derivative of it. And in this case, an acetyl amide derivative. Foods with glucosamine or other amide sugars will measure nitrogen and therefore influence the apparent protein value, just like other kind of non-protein nitrogens. Interestingly, fiber methods will capture these as well as a non-digestible carbohydrate, since the residue will remain after digestion, but as part of the fiber. Now, part of fiber methods is to subtract for protein residues. During the analysis, the residue will be tested for nitrogen content. And again, based on an assumed 6.25 factor, we're going to subtract for an assumed protein in that residue. Now, that factor of 6.25 isn't exactly correct for an acetylglucosamine either. So this makes interpretation complex for both proteins and fibers. Now, seaweed and algae also contain high levels of non-protein nitrogen, including sources of various amino sugars, and even chlorophyll is quite rich in nitrogen. Often when with upcycled and unique foods, there are surprises. I would suggest understanding the composition of novel foods and upcycled foods to fully understand the protein, amino acids, and non-protein nitrogen sources. Okay, let's move to protein declarations. As mentioned, the protein content can be declared based on the standard factor of nitrogen times 6.25 or other factors in official methods where they exist. For example, N times 6.38 in milk, N times 5.7 in flour, or N times 5.5 in gelatin. But when it comes to reporting the percent daily value, proteins differ from all other percent daily values. Other nutrients are always directly calculated without correction. So in this example here, we have a product that declares 4 grams of added sugar. Now, added sugars has a daily value of 50 grams, same as proteins. And that 4 grams is calculated out from the 50 to give you an 8% daily value. It doesn't matter if this is sucrose, lactose, fructose, or any sugar counts the same. But for proteins, if it is declared, it needs to be based on the corrected value. And it does matter what that protein is as described in the CFR excerpt. Let's look at these 3 examples. In each product, we have 21 grams of protein, but each is declared differently with the percent DV. The first is at 28% DV. This is an incomplete protein. That's fine. It still contains 21 grams protein, and that 21 grams is 28% of the daily value, which may be less than what you expect if you're trying to fulfill your daily value equivalent to 50 grams. Now, the second declares nothing. That doesn't mean that it's a bad protein. It just means it's not declared. And for many foods that you don't make a specific protein claim, you don't need to percent daily value. It is totally optional in most cases. There are specific requirements where it has to be declared. For example, if any claim is made, or if it's for foods for children under 4, then you must declare it. Now, this final example, 21 grams, 42% daily value. This is an example representing a complete protein, a PDCAAS of 1, and is the maximum percent daily value from that many grams of protein. Again, this is based on 50 grams daily value for adults and children 4 or more. If a product makes any claim related to protein, besides simply listing the grams of protein in the nutrition or supplement facts, it must consider that corrected protein content. Claims may be great source of protein, protein for muscle building, protein-rich diet, etc. Claims are made, you need to measure the quality of the protein and report that as a percent daily value. Now, an excellent source of a percent daily value is based on 20%, but that is based on the reference amount customarily consumed, or the RAC, which may differ from serving size. Good source, which is the minimum for making claims, is based on 10%. Let's look at these 3 example products. First, we have this Ancient Grains protein chips, making the claim of 10 grams from plant protein. We can make that claim, even if the proteins are not great quality, as long as we report the percent daily value in the nutrition facts panel, and the claim should correspond to at least a 10% daily value, which would be equivalent to 5 grams of complete protein. In the second example, we're not claiming any grams of protein, but we're still claiming a functionality based on its protein. So this should also list a percent daily value. And finally, we have this bone broth. Very popular today to have products with collagen, hydrolyzed collagen, bone broth, similar products. But sometimes there can be problems with how those claims are made. The 20 grams of protein may be truthful, but the percent daily value may be as low as 0% if this product is lacking tryptophan. An appropriate disclaimer on the quality of protein should be made. There are risks of class action lawsuits and regulatory actions against companies depending on how those claims are made. I often see products claiming, for example, 40% daily value from 20 grams of collagen or bone broth or gelatin. It's likely the manufacturer is not understanding the need for a PDCAAS corrected daily value. So let's talk about PDCAAS. It stands for Protein Digestibility-Corrected Amino Acid Score. Let me walk through a quick example, and then we'll get into more details. PDCAAS considers both the protein digestibility and amino acid score. So let's do an example that contains 7 grams of crude protein, and we determine 0.79% digestibility, 0.8 amino acid score. Those just multiplied will give you the PDCAAS value of 0.63. Now, with that 7 grams of crude protein, we can calculate a complete protein, which again is just a multiplication of those to give us 4.41 grams of protein. We do not declare 4.41 grams on the label. The label is still going to say 7 grams. It contains 7 grams of protein, but instead of declaring a 14% daily value, which we would if the PDCAAS was 1, we're rather going to calculate the 4.41 divided by the 50 grams to come up with a 9% daily value. And that's what's going to be on the label. That 9% is too low for claims. So if you want to make a claim, you would need to add more protein or find a higher quality protein. Let's dig further into those calculations. We can use digestibility factors as originally published in the Federal Register. This is our primary source for standard protein sources. There are other digestibility references. We have a number of resources to find this data. And at times, customers will come to us with already established digestibility factors for their products. If one does not exist, the official method is referenced here and is actually a live rat feeding study. It's not something that we do in my lab, but there are options to have a feeding study done to determine the digestibility. Final option is an in vitro digestion model. Now, this is not an officially approved by FDA, so there are some limitations on where I would recommend using it, but it is an option for quicker animal-free testing, which has been validated against animal models. Now, going back to my example where we calculated a percent digestibility, we need to know the sources of the protein. So when samples are submitted, we will ask, if you do not provide, what are the sources of your protein? If it's just one source, if there is multiple sources, and if it is multiple sources, we need to know the relative percent of each. Now, in this example, if someone were to provide, this is a combination of oats, brown rice, wheat flour, we would take the percentage of each, consider the digestibility of each, and we would come up with a weighted digestibility factor. Now, for the amino acid score, we need to test the amino acids in the product. For the 19 common nutritional amino acids, we can follow an amino acid analysis using hydrochloric acid with specific additives to prevent losses to certain amino acids. Then we use multiple derivatizations to make the amino acids selectively detectable by HPLC fluorescence or UV detection, using internal and external standard calibrations. This gives us data for everything except for tryptophan. In a second test, we can hydrolyze with sodium hydroxide, so a basic hydrolysis. This protects the tryptophan, and again, go through a derivatization and measurement just the same to give us a tryptophan result. Now, these two tests can give us the full profile of amino acids, but for the purpose of PDCAAS, we're interested in specific amino acids and how they compare to an ideal profile. So this schematic kind of shows more detail of what we're doing. We're starting off with long chains of proteins through digestions of HCl or sodium hydroxide. We are splitting those protein chains up into free amino acids, and through chromatography, we're sorting them out into piles. You can see how these different amino acids compare. The lysine and tryptophan are not filling their proportional cups. These would be examples of amino acids lacking versus some minimal requirement, while histidine here is overflowing. This is something that exceeds the requirements. That extra has no impact on protein quality. It is truncated at a 1 maximum. Of the 20 standard amino acids, 9 cannot be synthesized by humans, and these are listed here. Now, cysteine and tyrosine are also considered in minimal requirements as they are related to methionine and phenylalanine and can be converted into those, so the minimal requirement allows them to be summed. Those are amino acids that are considered conditionally essential. So the amino acid score is a comparison to the requirements of growing young children ages 2 to 5, really the highest level of need for amino acids. And in this table, we have the minimum requirement listed as milligrams of amino acid per gram of protein. If you exceed all these levels, that's considered a complete protein. Now I'm also comparing to my example product. You can see the milligrams per gram in my example product. If we do a ratio of these, we can find the ratio of how our product is versus the standard. Now the lowest ratio, in this case, this tryptophan here at 0.8, is the limiting amino acid. And we're going to only consider that lowest product as the limiting factor for our PDCAAS calculation. So again, we got a 0.79 or 79% digestibility. We're going to use a 0.8 ratio of the amino acid score to give us 0.63 PDCAAS value. We'll report that on our C of A. Then if for the grams of protein, we've measured 7 grams of crude protein. We consider the PDCAAS value, and we won't report the grams of complete protein, but based on that 4.41 grams and the daily value, we can report a 9% daily value. And that would be the value you could report on a label. So you may be asking, well, how do I improve protein quality? Many plant-based foods by themselves have lower PDCAAS values, but there are complementary combinations. Rice and beans is the classic common combination in many diets, especially in regions where meat is less economically available, and for good reasons. Now, grains lack adequate dietary levels of lysine, but may have sufficient levels of sulfur-containing amino acid methionine, while legumes are generally higher in the essential amino acid lysine, but they're lower in that sulfur-containing methionine. Similarly, corn and soy are often combined, as soy has lysine and tryptophan, which are deficient from corn. So depending upon the product, there may be ways to improve a PDCAAS. For general populations, protein quality is not emphasized in FDA or the USDA dietary guidelines for Americans. Generally, adult humans eat a varied diet, and lacking amino acids of any one meal or a component of meal is not that big a concern. We will likely make up for it across multiple meals. PDCAAS is based on the worst case growing child requirements. Most Americans are not amino acid deficient. So FDA does not require percent daily value in most cases. It's just when you're making claims, then a product needs to stand up to a higher standard. Now, free amino acids can be added to foods to significantly improve biological quality of total protein within some limitations. Generally, dietary supplements will contain specific amino acids, which alone cannot be used to make protein claims, but may be able to make other claims to their structure function, such as branched chain amino acids for building muscle or exercise recovery. Free amino acids should not be counted in the grams of protein, but they can be considered in the overall product in the PDCAAS. Novel foods like yeast, mushroom, upcycled and fermented foods will vary in their protein quality, but through research and optimization, you can improve amino acid scores, and it's possible to also pursue a digestibility study that may be needed to determine its specific value to make a claim. There are other measurements for protein quality. I'll just mention protein efficiency ratio, PER. This is a very commonly used method as well. It is the official method for infant formula, and it's also the official method in Canada. Now, Canada also recognizes an allowance for making a conversion factor to PER. So PDCAAS values are generally allowable to use for protein claims in Canada. Now, PER is a rat feeding study specifically measuring the weight gain under specific conditions. Next, I'll mention DIAAS, Digestible Indispensable Amino Acid Score. Now, DIAAS has been promoted by the FAO, World Health Organization, as a better measurement to PDCAAS. And it's based on the true ileal digestibility of each amino acid, which means how the amino acid is absorbed prior to the colon, therefore excluding colon fermentation. Now, this is preferably collected in humans, but you can imagine it's quite an invasive means to collect feeding data. Pig studies are also recommended as closer to humans than rats, but regardless, all these are quite difficult, and the methodology around this has just not reached common use. I need to make a few comments about calories from proteins and related ingredients, as there often is some confusion and questions about this. There are standard means to calculate calories, some of which are listed on this slide, with 4 calories per gram from protein as being most commonly used. Bomb calorimetry is another method with an established correction due to incomplete digestibility. But what about non-protein nitrogen? Do the regulations address calories from them? Well, you should keep in mind that the framework of calorie calculations is very old. Wilbur Atwater studied nutrition and metabolism and the foundation of caloric factors in the late 1800s. This research and subsequent research was on human diets and the foods eaten in that time. So it's not a surprise that the rules established so long ago can be challenging to fit into the foods, dietary supplements, and trends of today. Atkins or keto diets would have been a very foreign concept to the nutritional founders. Labeling of dietary supplements is also a concept that came much later in time. Following the simple crude protein calculation and carbs by difference with 4 kilocalories per gram of protein or carbohydrate, regardless of which it fits into, is the easiest and safest approach. But marketers will often want to minimize calories and look for ways to exclude components in a calorie calculation. Free amino acids should not be counted as protein, but that does not specifically mean that free amino acids are non-caloric. From a scientific view, amino acids are of course caloric. Energy from protein is only possible from the digestion of protein into amino acids. Now, guidance on this topic is lacking from FDA. However, there is an example in the CFR that clearly shows about 4 grams of protein, if you add all these together, of free amino acids and a declared value of 15 calories, which is just exactly as what you would expect from measuring these free amino acids as 4 calories per gram, or even a bomb calorimeter. Many other components of foods don't fit neatly into the narrow definitions of protein, fat, or carbohydrate, but in many cases, they actually do have caloric content. So for our labeling, unless we have specific reasons to exclude, non-protein nitrogen is generally counted just like proteins as 4 calories per gram. Let's move on to some targeted protein analysis. This is primarily for claims on specific functional foods or dietary supplements. Among some of the methods we offer is lactoferrin. This is achieved through a selective peptide digestion with internal standard to selectively ID the peptide pattern and quantitate on a high-resolution mass spec. Similarly, alpha-lactalbumin, beta-lactoglobulin are measured against specific peptide digestion. Are you familiar with A2 milk? A2 milk has a beta casing that differs by 1 amino acid from A1 milk and may be tolerated differently by some individuals. We can support A2 claims by testing for the A1 beta casein and the total casein. Again, by selective peptide digestion, that allows us to measure for the A1 as a qualifier for the purity of the marketed A2 milk. Now, some methods will allow for a simpler HPLC UV measurement. There are examples of standardized methods like immunoglobulin G in milk or colostrum, where a validated method to industry standards is available. Now, collagen is something that we primarily address through a hydroxyproline analysis, according to the same amino acids analysis described earlier, and using a factor of hydroxyproline to convert to an estimated collagen content. It is possible to measure collagen peptides, and we have some options, but separation and specificity are challenges. There is a real need for more standardized methods to support claims of today's products. We do have a number of proprietary projects in the realm of precision fermentation. There's much interest in identical to animal-based proteins through fermentation, as well as many other functional proteins. Characterization is needed for GRAS dossiers, shelf life, quality control, and research on products. We can help develop methods for the identity, as well as to measure the purity or assay against a reference material. I'm going to finish with a brief overview of other protein characterizations. Some of these techniques are available in the Madison lab, but others are part of our wider network. SDS page or electrophoresis can be used for molecular weight separation or to separate common protein types, for example, casein or whey. HPLC UV methods are commonly used for peak area purity in relatively pure protein products or assay concentration when a standard material is available. Now, high-resolution LC mass spectrometry can be used to measure the mass, and through partial enzymatic digestion of peptide sequences, we can use it for identification and very accurate quantitation. Often the simpler HPLC UV method can be developed alongside of a high-resolution mass spec method so that you have a simpler purity method for more routine analysis and a generally a lower cost, but it is supported by a very specific high-resolution mass spec method, which has data to provide the identity of the peaks. Now, GPC-SEC, or gel permeation chromatography, size exclusion chromatography, can be used for hydrolyzed molecular weight distributions. ELISA, or immunoassays, are very commonly used for allergen assays. They're very specific to the allergen part of proteins, but they can be used for other purposes to measure a whole variety of targeted proteins. Degree of hydrolysis, the OPA method, is a way to measure relative hydrolysis as well. This relies on a reaction with primary amines to form a chromophore and UV spectrometer reading. It's relative based on the protein type because side chain reactions affect the measurement as well. Now, selective precipitation is the basis of some standard methods, especially in the separation of casein and whey. We know that casein and whey will behave differently at pH 4.6. So by precipitation, we can separate them and measure by kind of standard crude protein measurements. Finally, PCR methods are also available tools to go along with the protein test. One example is to measure for the DNA as a presence of meat, milk, or egg DNA in vegan products as a contaminant. These products are typically run on processing lines that may have milk, meat, and egg. If you want to verify that the absence in a vegan product, DNA will give you a more simple way to measure that. PCR is a more robust way to measure for the presence of GMO in meat proteins, basically just because of how proteins denature, how there are differences in proteins. It's more efficient and robust to measure by DNA PCR techniques. Lastly, for development work or to troubleshoot product performance, there are many other ways that we can characterize kind of beyond kind of standard chemical analysis, the more functional analysis. This can be measurements of solubility, pH and heat stability, water holding, emulsion and foam capacity, and the stability for each of those measurements. Also, we can make measurements for viscosity and gel strength, and lastly, particle size measurements. These measurements are usually for relative comparisons, not necessarily routine analysis. It's usually part of a project to support at some kind of end goal. I've kept the details pretty limited in this characterization because, as you can see, there are many options and each can get pretty complicated. But my colleagues in Magnuson and other sites can discuss in more detail and propose a project to meet your specific needs. Well, with that, I thank you for your attention, and I can have some time to take any questions.
All right, great. Thank you so much, Dan. We have a couple of questions here. So the first one is, what do I do if I'm unsure of what protein testing method is best for my product?
Okay, let me guess answer that in two parts. So you're unsure of what testing is for your product. You know, as I went through in the presentation, there's a number of questions that you need to ask as far as the purpose of the testing. So, are you interested? Is this for a claim? Is there other sources of nitrogen? Are there non-protein nitrogens in your product? Are you interested in a very specific protein? Those are kind of the foundational questions that will help guide, do I just need a crude protein analysis? Do I need to do a crude protein analysis in combination with a measurement for carnitine, for example? Do I need to do a non-protein nitrogen analysis? or am I really looking for something very specific and I need to choose a specific method? The second way I'll answer that question is really about finding the answers, right? So there are a number of ways that you can interact with Eurofins to get answers to your questions. We have a network of client service representatives, business development, a number of subject matter experts. You know, if you have a contact that you're working with, you know, those are kind of your first point of contact for questions. Some of these questions can be directly answered by just reviewing information through our EOL, or Eurofins online portal. Most of these methods will have a data sheet that will provide some information which may answer those questions, as well as our website. Our website will have you know, this particular webinar, but other webinars, as well as some other documents of white papers, infographics that will give you, you know, information that may help answer these questions and maybe a good reference ongoing. But again, you know, as you can feel free to ask your client service representative or other contacts for help in making these decisions, because they are complicated.
Great, thanks, Dan. We have another question here from the group. What do I do if I use multiple protein sources and want to claim a % daily value on my label?
Right. Okay, so if there's multiple protein sources, we need to consider that in the protein digestibility. It doesn't matter for our amino acid score. We're gonna, regardless of all the sources, we're gonna measure for amino acids all the same. But for protein digestibility, most typically we're using a factor calculation. So we need to know what types of proteins, and ideally, you know, a little more about the proteins. For example, if I'm eating whole wheat versus a wheat flour, there may be some difference in that digestibility score. So what we'll ask for, if ideally, if you can provide in your order, if you don't, we'll ask for it is a breakdown of the different sources of those proteins by %. Ideally, you would provide, say, this product contains 50% of its protein is from milk powder, 30% is from dried eggs, and 20% is from a dried yeast powder or something. You know, again, if we get information, if you don't know exactly that, but you have formulation information and you have specification information for those ingredients, we may be able to infer that. Again, we may not need to know 100% of the formula. There's going to be some very minor ingredients that may contribute to small amounts of protein, maybe from flavors and such. So as much information as you can provide, we'll do our best to make an accurate assessment of that digestibility.
All right, perfect, thank you. And then we have time for one other question. Can you clarify if there are options to add amino acids to improve protein quality for a protein claim in a food or dietary supplement?
Yeah, that's a good question. So you can. It needs to be a protein that is, you know, specifically low. So you know, if you have a plant protein that is specifically low in its PDCAAS, there are allowances. Again, this is dependent on the amino acids being grass, you know, food, grass additives, well, not food additives, but grass allowed ingredients to add. And then the PDCAAS would be, again, measuring for the crude protein, the amino acids that have been added to the product and declared within the ingredient statement would contribute to the final you know, amino acid score and make an improvement.
Okay, great, thank you. Well, thank you, Dan, so much for today's insightful presentation, and a big thanks to everyone who attended. As Dan mentioned, please reach out to the Eurofins team with any additional questions. We definitely would be happy to connect further to discuss, this important topic along with other topics. This now will conclude today's webinar. Everyone, please have a great day.